Hey everyone, welcome to part 2 of our continuing discussion on vision. This time we're talking about low-level processing, stuff that happens before we even get into the brain. Let's start by establishing some terminology. A receptive field. This is the portion of the visual field, so everything that you can see, where a visual stimuli will produce an alteration in the firing rate of a particular neuron. So a receptive field is just the portion of your visual field that one individual neuron is responsible for. We can talk about the receptive field of a photo of one sort of individual rod or cone, or receptive field at the level of a ganglion cell, which will make more sense in just a second. Acuity is determined by ganglion cells, right? So the ganglion cells in the fovea that receive input from fewer receptors than those in the periphery have sharper acuity. Basically, the more condensing of a signal you have in terms of the number of receptors that are talking to a single ganglion cell, the worse the acuity is. So in the center of our receptive field in the fovea, we have a really high level of conservation of information, right? We have only a single photoreceptor that is talking to a single bipolar cell that's talking to a single ganglion cell, right? So a high level of conservation. However, in the periphery, we have a large number of receptors that are talking to a smaller number of bipolar cells that might be talking to a single ganglion cell. So you can see that in the periphery, we have a lot of receptors being condensed to a single ganglion cell, so a loss of acuity as we have a lot of information condensed to a single processing unit. In the fovea, we have greater acuity because we have fewer photoreceptors being condensed down to the level of one ganglion cell, right? Okay, next let's talk about the coding of light and dark that happens at the level of your ganglion cells. This can be a little confusing, so we'll talk through it in a couple of different ways. So you can think about your receptive field consisting of a roughly circular center surrounded by a roughly circular ring, so sort of a concentric set of circles. These are what we would call on-off ganglion cells and a center surround sort of organization. So what we'll call on cells have an excitatory center portion and an inhibitory surround portion. So firing increases when light touches the center of the visual field, and firing decreases when light touches the surround. So off cells, on the other hand, have the opposite organization, where stimulation of the center portion of the visual field will produce inhibition, so decrease firing of the ganglion cell, and stimulation in the outer ring will be excitatory. So on cells have an excitatory center and an inhibitory surround. Off cells are just the opposite, inhibitory center, excitatory surround. So looking at that all at once, here's kind of what we would see, right? If the center is being stimulated by a point of light and the surround isn't, we would see an increase in firing. If we see the outside of an on cell being stimulated, we would see a decrease in firing relative to baseline. And just the opposite for the off cells, right? In the center, we see a suppression of firing, and in the outside ring, we see an enhancement of firing. Okay, so let's look at this in a little bit different of a way. So here I've got a sort of highly simplified and highly schematized version of an on cell with an excitatory center and an inhibitory surround. So this right here is sort of meant to represent that. This is not a real physical thing, right? There's not like a disc floating above your rods. This is meant to represent the organization. So these two photoreceptors are going to represent the center, the on portion, and these two photoreceptors on the outside are going to represent the outside ring or the off portion because we're talking about an on cell, right? So those are the photoreceptors. In this space right here, we're going to represent the influence of bipolar and horizontal cells, right? We're really kind of glossing over this. We're not going to go too much into the weeds about how exactly bipolar and horizontal cells can modulate this. I'm just going to represent it with basically an excitatory inhibitory arrow. Then we have our ganglion cell, which is going to produce our output in our optic nerve. So let's start with something simple. A little bit of light comes down and stimulates the center of our field, right at the on portion of this field. So it's going to activate these two photoreceptors right here, which are then going to, via the bipolar and horizontal cells, send an excitatory signal down to our ganglion cell, which is going to fire. So we can see here during the portion, if we sort of think about this as a crude representation of our membrane potential, Every time you see one of these perpendicular lines, you can think about the ganglion cell firing and action potential. You can see that during the period of time when our light stimulus is present, we see an increase in the rate of firing of our ganglion cell, right? The excitatory portion is being stimulated. These photoreceptors are firing because those are the excitatory photoreceptors in the center of this field. They're going to influence the ganglion cell to increase its firing rate. Okay, so using the same sort of layout and the same type of cell, let's look at a slightly different kind of stimulation. So what if we have a broad stimulation where light hits both the on and the off portion? Well, what happens then is all of our photoreceptors are stimulated and we get a mix of inputs, right? The inhibitory receptors on the surround send inhibitory signals down to the ganglion cell via, of course, the bipolar and horizontal cells, while the ones in the center keep doing what they were doing before with an excitatory signal. What we see there is sort of a more modest increase in the firing rate as it's sort of dampened by the inhibitory circuit. Okay, for a third example, what happens if light just hits the outer ring, the inhibitory portion of this receptive field? Well, then only the photoreceptor on the outside here, on the right side, that is part of the off or inhibitory surround is going to be stimulated, which is going to influence our ganglion cell in a different direction. So that's actually going to cause suppression of firing, right? So while the surround is being stimulated by that little beam of light, we see that the baseline firing rate is completely suppressed, and we see no firing while this light is present. Interestingly enough, we see a little rebound, right? So after this cell has been quieted for a little while because of the influence of our photoreceptor input to the ganglion cell, after that light stimulus is removed, we see a brief rebound where the ganglion cell sort of bounces back and fires at a higher rate briefly. Okay, so what about the other ones? We talked a little bit about the off cells with an inhibitory center and an excitatory surround. As you might expect, if we have a beam of light targeting the center here, the off portion, it's going to stimulate these photoreceptors in the center, which are going to exert inhibitory influence over the ganglion cell. So we see the same thing we saw in the previous example, right? The stimulation of the center of an off cell is going to cause suppression and then a brief rebound. Likewise, in the same off cell, if the excitatory periphery is excited, we have the same sort of pattern we saw before where this one photoreceptor is able to exert influence over the ganglion cell. In this instance, we have an increase in firing, right? The excitatory outer portion is being stimulated, which is driving an increase in firing in our ganglion cell. So you might be wondering, why in the world do we have this complicated system at our receptor level? Isn't this something we can handle later on in the brain if we need to do it at all? But one thing that the really cool thing that the organization of our ganglion cells lets us do is the enhancement of contrast, right? So if we have a bunch of on cells here, you can see that there's sort of a line that's formed. So if the cells, the on cells in the light portion are receiving like light stimulation, and in the dark portion here are not receiving light stimulation, like there's a cutoff of where our stimulus is, you can see that these cells are all evenly illuminated. So we have that sort of modest firing rate that we talked about before. But here on the edge, you see that only a portion of the inhibitory surround is being illuminated. So if the outside is what's exerting inhibitory influence and it's not as strongly stimulated, then we're going to have a higher rate of firing in the ganglion cells that are supporting these fields, right? Because there's less inhibition, there's less activation of this inhibitory surround. Less inhibition means a higher firing rate, which means we actually see this as a brighter edge, right? We have an enhanced contrast. We can tell where the light ends and where the darkness begins because of our ganglion cells. So if you look at a stimulus like this one, a gradient, we can have enhancement of contrast here. The center surround organization will enhance our ability to detect edges. And detecting edges is actually really important for survival in a lot of situations. So I can tell you now that each bar is uniform in color, but one edge will appear lighter and the other will appear darker. So if you look closely at these borders between the light and dark, you can see that there's sort of this artifact where the side on the right here might appear lighter and the side on the left appears darker. This is the same thing that we were showing here on the previous slide. Because of the uneven sort of illumination of those border ganglion cells, we see an enhancement of those edges. Okay, so we spent a while on that. Let's move on and talk about how we code visual information. So We have color vision, as you all know. Objects in the environment selectively absorb some wavelengths and reflect others, right? So depending on what wavelengths of light an object is absorbing and what they are reflecting influences what color we perceive them to be. We have what's called the trichromatic theory of vision. This is proposed that color vision was possible due to 3 different receptors, each responsive to a different hue. So we have red cones, green cones, and blue cones. The cones aren't physically that color, it's the wavelength of light that they respond to. This was based on the observation that any color can be reproduced by mixing those 3 colors. So the reason we can look at our lovely school icon and see this variety of wonderful colors is because of the contributions of our 3 cone types. So looking at this a different way, the idea is that each of these 3 cones responds preferentially to a specific wavelength of light, or a range of wavelengths of light. So blue cones are at peak sensitivity to 419 nanometer wavelength light, so shorter form light. Green cones, 531, and red cones, 559. So the idea is that we have these different cones that respond at peak to these different wavelengths of light. They're sensitive to a wider range, but this is the peak responsivity. This is caused by actual physical differences in those cones. They contain different opsins, right? That photoprotein that is sensitive to the presence of different wavelengths of light. So because different opsins are contained in each cone type, they are differentially responsive to different wavelengths of light. Color blindness is actually attributable to differences in cones between people. So protanopia is when red and green hues are confused, both appear sort of yellowish, where red cones are filled with green cone opsin. This is the most common form of color blindness. Deuteranopia is also confusing of red and green hues. Green cones are filled with the red opsin. So in somebody with red-green color blindness caused by either form here, this is the sort of colors that we might see with normal vision, and these are how those colors might be seen as somebody with a red-green color deficiency. Tritanopia is much more rare, where an individual lacks blue cones, and they might see things like a bright green sky, or yellow might appear to be pink because of the absence of those blue cones. So in a somewhat similar way to when we were talking about light and dark, at the level of the ganglion cell, these ganglion cells respond to primary colors. So they're organized in a similar fashion to what we talked about previously, with an excitatory center and an inhibitory surround. But instead of talking about light and dark, this time we're talking about color. So ganglion cells respond to pairs of primary colors. So red versus green, yellow versus blue, etc. One of the reasons why you can't imagine a color like a reddish green is because you are physically incapable of perceiving something like that, right? You see either red or green. You cannot see with the same visual field red and green at the same time. If you're seeing red, your ability to see green is suppressed. If you're seeing green, your ability to see red is suppressed, right? Because of the the opponent process nature of your ganglion cells, when you're seeing red, you're actively suppressing the ability to see green. If you're seeing green, you're actively suppressing the ability to see red. And that's one of the reasons why we can see a negative afterimage. If you remember when I showed you previously with the light-dark example, when your visual system is briefly suppressed, it will experience a rebound period where the suppressed functionality will begin firing at a higher rate. And your ganglion cells, to do with color, do the same thing. I'm going to show you an example. So what I'm showing you here is just a box filled with 4 sort of bright primary colors. What I want you to do is stare at this little black dot in the center of the figure. Go ahead and pause the video right now and stare at that black dot for about 30 seconds. Keep your eyes focused on the black dot and don't let them move around. Just focus on the black dot and let those colors sort of fall on your retina. Okay, go ahead and pause the video, do that for about 30 seconds, and then move on to Okay, so if you do that correctly, you should be able to see an afterimage of sort of the opposite of the colors that you saw on the previous slide. This isn't a trick, this is just a blank white square. So the colors that you are seeing are completely in your own mind. Well, I mean, I guess all colors are completely in your own mind when you think about it, but that's not what I mean. What you're seeing is the sort of rebound effect from what you had from these cells that you had fatigued, right? By suppressing the opposite of the color that you were seeing, now you're having a rebound effect where you're seeing the opposite of that color in the absence of any sort of stimulation, right? There's no colors in the square, it's a blank white square. So just to sort of reiterate, right, when you're staring at this little fixation marker and this portion of your retina that's seeing blue is being stimulated with this blue light, when that's happening, it's turning yellow off, right? It's suppressing your ability to see yellow light. So when you remove this stimulus, you see yellow here as those yellow detecting cells rebound and produce the sensation of being able to see. So here's another example of a negative after image. If you look at this inverted American flag, right at the fixation marker here, and then transfer your gaze to this fixation marker, you will see an American flag in the normal colors. So this is caused by adaptation to firing rates, right? So for example, blue light inhibits yellow or sensitive cells. The visual system will adapt to that level of firing. And then when the blue light ends, the yellow shells show a rebound effect. They fire faster than the normal baseline, and it causes us to perceive yellow, despite there being no yellow present. Okay, that's it for our discussion of basic processing. Next time, we're going to move on to a discussion of the LGN of the thalamus.